Assalamu alaikum. Welcome back to lecture 3 of DNA and design. So now we're actually ready to talk about DNA itself and some of the issues that suggest, at least to some of us, intelligent design. To do that, we actually need to get a little bit oriented to the structure of DNA and how it works inside the cell. And I know that biology is far away for many of us. So we're going to have to go into a little bit of detail. I will ask you to bear with me because if we want to find pearls, we have to dig for oysters. Or dive for oysters, or whatever the appropriate metaphor may be. But it will take us a little bit of effort, but I really do believe, inshallah, it will be worth our while in terms of the payoff and understanding. And after all, this is the genetic code that passes information from parents to children. This is the basis of heredity. So it's probably a good thing for us as informed Muslims to have some idea about. And certainly the level of detail we will go into, even though it may seem like unnecessary detail, is really only a minimal scratching of the surface of what has been discovered about the very complex world of genetic control. So when Watson and Crick finally discovered the structure of DNA, they found that it is basically a double helix molecule that is made up, as the name suggests, of deoxyribonucleic acid. And what does that mean? It means that there are 4 chemical DNA bases, and they are guanine, cytosine, adenine, and thymine, and you can see them diagrammed in the picture on your right-hand side. Some of these bases have a double ring structure, and these are guanine and adenine. Some of them have a one ring structure, and these are thymine and cytosine. And the structure of DNA is basically that a base, one of the 4 bases, G, C, T, or A, as we will abbreviate them from now on, is hooked to a 5-ringed sugar, or a 5-carbon sugar, known as a ribose. And that in turn is hooked into a phosphate group, an atom of phosphorus linked to 3 oxygens. And the phosphate and sugar portions form the so-called backbone of the DNA. And from that, sticking inward are the bases. And the bases then bond to each other through hydrogen bonds. And it turns out that they pair in a very interesting way. That guanine can only pair with cytosine, and that adenine can only pair with thymine. And so I have the 4 DNA bases on the inside hooked into a backbone made of a 5-carbon sugar, ribose, and a phosphate group, and that backbone spirals around into a sort of helical strand, and I have one helix on each side that bind together through the hydrogen bonds that bind the DNA bases. G binds to C and A binds to T always. Now, when we come to talk about DNA replication, because you may have wondered, okay, how do I pass genetic information on? When cells divide, we know that cells divide as the fetus grows. We know, for example, that cells constantly divide in most of our body to replicate damaged cells. Basically, because G can only hook to C and A can only hook to T, imagine if I only had one helix of the double helix. So looking in this particular molecule at starting from the top, I have G, T, C, and A going down. Well, if I only had that half, I know what has to hook to the other side because G can only hook to C, T can only hook to A, so I can then tell you that the other helix has to be C, A, G, and T. And that is precisely how the body replicates. It peels the strands apart and then hooks the complementary strand, makes a complementary strand for each of the two strands, and we will cover that in a little bit more detail later. I just wanted to preface it for you. But now we understand the basic chemical structure of DNA. Okay, so knowing what we know so far, we can make some pretty amazing statements. First, DNA stores the genetic code using a 4-letter alphabet, and that is the 4 base pairs we talked about, G, C, T, and A. Just like in English, we communicate information using a 26-letter alphabet. Well, the DNA, which is stored in the chromosomes of our cells, uses a 4-letter alphabet to convey all of the genetic information that makes us live, makes us synthesize proteins which do the work inside the cells. And the code of all of the proteins is determined by the order of the DNA base pairs. So the way I order those letters determines the structure of a protein. Just like the way I order letters in English determines the actual words that are then made that convey a certain meaning. And if I change the order of the letters, I would change those words or that meaning. The sections of DNA that code for proteins are called genes. And so a section of DNA which codes for a protein that performs a function in a cell, that is called a gene. And some genes determine eye color, some genes determine blood type, and so on and so forth. Now, it is important to note that DNA and modern genetics were entirely unknown to Charles Darwin when he came up with his theory of evolution. He said that random changes in organisms were acted upon by natural selection. For example, a giraffe that had a slightly longer neck than other giraffes might be better adapted to survive, and we start then selecting for giraffes with slightly taller necks, and over many generations we evolve an animal that has a very long neck, i.e. a giraffe. But it was unclear what would cause one giraffe to have a slightly taller neck? What would cause a bird to have a slightly more pointy beak? What would cause a butterfly to have a slightly different coloration? Well, that is differences in the genes. Okay, so in just a few minutes, we have learned an awful lot, and we will keep delving into some of the details of the structure of how DNA does what it does in the cell, because it will become important for us as we move forward. Okay, so now we want to touch on two of the main functions of DNA in the cell. The first is replication. DNA has to be able to replicate itself, whether it is to grow new cells, like for example new blood cells or new skin cells or new liver cells in the organism, or to pass on the genetic information to the next generation through the gametes, through the sperm and ova, for example. And so we've already sort of covered how DNA replicates itself. We'll get back to this topic in a little bit more detail later, inshallah. But the idea is that the double helix unwinds into two separate strands. And because each base has a complement, a specific complement, G can only bind with C, T can only bind with A, then each of those single strands starts to collect new DNA bases to complement the strand that already exists. So if I peel the strands apart, as you see in the diagrams on the left, then enzymes start bringing in new DNA bases and hooking them up to their complements on the strand that I already had. And that way, each strand now becomes its own double strand, and we can replicate the DNA molecule. So that is DNA replication in a very, very cursory fashion. As I said, we'll come back to this point in just a little bit more detail later, inshallah. Okay, so we said we would look at two main functions of DNA. One is DNA's ability to replicate itself for our cells dividing in our bodies and for passing on genetic information to the next generation, and we have covered that. The next important aspect is, how does DNA make protein? You can think of the main function of DNA as making protein, or specifying the information to allow the cells to make the proper proteins that it needs to sustain life. So how does DNA do that? Well, before we get there, let's just do a very brief review of what a protein is. A protein is basically a chain of amino acids. Amino acids are chemical compounds that are a combination of an amino group, something called a carboxyl group, and a side chain. All amino acids have the same amino group, they all have the same carboxyl group, but they have different side chains. And it is the different side chains that distinguish one amino acid from another. In living cells, in us, for example, there are 20 different amino acids which are used to make proteins. And those amino acids are linked to each other in a long chain. Think of it as sort of beads on a string, sort of like a pearl necklace. And then once those amino acids have been linked in the right order, that chain folds up in a complex way, and that forms a functional protein. So DNA carries the information to specify the order of amino acids that will go to make the protein. Okay, I hope that you are following with us so far. So the question now becomes, how does this happen? How does DNA convey this information? In other words, how does the four-letter alphabet specify the order of amino acids to make up a proper protein? You remember we said that Watson and Crick discovered the double helix structure of DNA. Well, a bit later, Crick was the one who unraveled or answered the question we are now asking. He basically discovered that the four letters in DNA are used to make three-letter words. These three-letter words are called codons, and each codon corresponds to a specific amino acid. So if you look, for example, at this slide, it is showing something called RNA. And so the first step is that DNA becomes turned into something called messenger RNA, and messenger RNA is actually the part that has the codons that correspond to amino acids, and we will flesh this out in the next slide. Okay, so this process is a little bit more complex than the replication we talked about, but please stay with me. Again, this is, the nature of life, if you will. Let's look at the top DNA molecule. As we said, it is made up of two complementary strands, and each strand is made up of the four-letter alphabet, the four bases, T, A, C, and G. And so we see that, for example, our top DNA strand, let's focus on that. That's the one that will turn into a protein for our purposes. It starts off TAC, GGC, GTT, and so forth. And you notice I am grouping the bases as I say them in groups of three, because it turns out that the four letters are used to make three-letter words, and those specify the order of the amino acids. How does this happen? Well, the first part is called transcription. The DNA strands peel apart, and in a process very similar to DNA replication, new bases are brought in to sort of copy the information. The DNA gets transcribed. So let's again focus on the top strand. It starts off TAC. So we would expect its complement, just as shown on the bottom DNA strand, to be ATG. Well, what happens is that instead of making a new DNA strand to make a protein, the body makes something called RNA, which uses almost the same basis as DNA. It has a slightly different structure that we'll talk about later, but instead of thiamine, it uses something called uracil. So anytime that a T would appear, we substitute instead a U. So the first three bases, TAC, then correspond to their complements AUG instead of ATG. The next three bases in the top DNA strand, GGC, now correspond to their complements CCG, and so forth. And each group of three RNA bases forms what is called a codon, and each codon corresponds to a specific amino acid, which you see on the bottom. So we see, for example, that AUG corresponds to something that says MET. That stands for the amino acid methionine. CCG stands for the amino acid proline, and so forth. So now we see that the four-letter alphabet of DNA goes to specify a complementary RNA, and that RNA is made up of also four bases, but instead of A, T, G, and C, it's A, U, G, and C. Those are the letters that make up RNA, and those letters are taken as three-letter words called codons. Each three-letter word corresponds to a specific amino acid, to one bead on the necklace that makes up the protein that we discussed earlier. Okay, so let's do a quick recap to end this lecture. I'm sure you're probably really can't wait for it to end because I threw a lot of information at you. But basically, the letters on DNA are transcribed to make something called messenger RNA, ribonucleic acid, and those bases or those letters on RNA, which you can think of essentially as a mirror image of the letters on DNA, are taken in terms of three-letter words. Each three letters form one word called a codon, and each codon corresponds to an amino acid. So in this messenger RNA, you see that the first codon is GCU. The second codon is ACG. The third codon is GAG, and so forth. Now, I had said earlier that there are 20 amino acids that make up the proteins in our bodies. Now, if we think about it, we said that each codon, each word, is made up of three letters. And each letter can be one of four possibilities, the four bases of RNA, A, U, G, and C. So the first letter could be any one of four possibilities. The second letter could be any one of four possibilities. The third letter could be any one of four possibilities. So how many possible three-letter words can I make from this alphabet? Well, it would be 4 times 4 times 4, which is 64. And you see those 64 here on the table on your right. So this sets us up for something interesting, because I have 64 possibilities to specify amino acids, but I only have 20 amino acids. So how does this work? Well, the way it works is something called redundancy. What that means is multiple words can specify the same amino acid. So if you look, for example, at the amino acid leucine, you see that CUU, CUC, CUA, and CUG, all of those words, all of those three-letter codons specify leucine. So anytime CUU or CUC or CUA or CUG show up in a messenger RNA, leucine is put into the protein. If you look a little bit further down in that same column, you see that there are also four different words that can specify valine. Now, if you look on the top right of the table, you see that there are only two letters that will specify cysteine. I'm sorry, two words that will specify cysteine, UGU and UGC. And in addition, we see that a few of these words mean stop. They are called a stop codon. What does that mean? It means that when they appear in the messenger RNA, the body knows, the cellular machinery knows, okay, stop here, we're done, stop adding amino acids. So for example, UAA, UAG, UGA, those are stop codons. And so now we have covered the genetic code, the language of all life. And so I know it was a lot of detail, but I hope it was worth it. And this now sets us up to be able to talk about how this system gives evidence of intelligent design. We'll see you inshallah in the next lecture. Assalamu alaikum and God bless.